All right, so I got some more R stuff to do, and I don't know if it's necessarily new. Some of this stuff will be new, but also some of the stuff that I'm going to talk about will be related to a little bit more a little bit more data manipulation, and just explanation of stuff that I've already done. Um, Weird. It seems like my chat's. I don't know if that's accurate or not, but whatever. Whatever. I think everything's fine. So I'm pulling up my old script that we wrote out on Wednesday, and so I'm gonna run this real quick. Source. Let's see. Uh, blah, blah, step. And so I'm just gonna run the script again, and then bring up my end drawing. It's Lin Hist. So I'm just going to bring this up because I haven't shown it yet. I've been meaning to. I keep forgetting to, but I just want to do this first is to actually save a. So in my figure, so whenever I get this figure pulled up, I do have this export button, and so I can save this as an image. All right, I know fucking a. All right, I think this is working again. I'm sorry. This isn't y'all. This is all me. It's like sometimes as soon as I want to do something, like my internet's all fine all day, and then as soon as I want to stream or talk to somebody, it's like, oh, I give up. Whatever. So if I have this figure pulled up, uh, my export button, it lets me save it as an image or a PDF. So a PDF can be manipulated in other software. So like Adobe, Photoshop, or Illustrator, that kind of tool, a PDF can be manipulated in if we want to do things further to it. But otherwise, we would just save this as a an image. So you can save it as a PNG, a JPEG, a BMP, whatever. They're all more or less the same. However, an SVG is editable as well as an EPS. Both of those file types are editable. But we'll just save this as a PNG. It's going to save this figure in the same directory as my source directory. So I'm just gonna rename this to this is blast link dot png. Again, making sure that my file extension matches the file type, and so that'll be saved. Um, and so we have it forever. We don't have to keep drawing this graph out every time that we want to have it. We just have it saved. All right. So I generated another data frame from some of my own work that I wanted to walk you through a little bit. If anything else, just to walk through some other tools in R that help manipulate data frames. So this one is this is related to transposable element expression. I've already shown you some data from this graph, but I simplified it a little bit further, make it a little bit easier because I will probably pass this out to you for y'all to play with. Just need to forget what it's called. So I'm going to call this dat2 read.table. What is this file called? Um, mouse t expression stuff. And so in this folder, in this file, I do have headers. So my header is set to true because I do have, my columns are named, and it is still separated by a tab. So I will define that. Sep is tab. It's going to complain I'm missing something stupid. I am missing blah. Oh my gosh. Stop it. Stop it. Stop it. That was stupid. Okay. Hey. So like day two. So if we're looking at this figure, or if we're looking at this data type, so I have my species defined. I have the type of I have the type of fucking A. All right, so I'm about to give up on this. Okay. Species defined, type defined. So my types are line, signs, LTRs, DNA transposons. These are all the big four categories of transposable element types. So line stands for a long interspersed element. Sign is a short interspersed element. LTR is a long terminal repeat, and LTRs are kind of very similar to a lot of viruses. Um, and DNA transposons, they're their own unique kind of TE. But anyways, also have specific families. So there's definitely more than one type of line, more than one type of sign. So they get defined into families. And also have how many insertions of this specific family are found in the mouse genome. So this specific line family, there are 400 different copies of this element in the genome. And then I have two metrics of expression. So this A mouse expression is the measured expression of this TE family in an adult mouse testis. And the J mal expression is the same thing. We're just looking at expression of these TEs in juvenile mouse testis. Um, because a lot of times transposable elements are expressed in every tissue. It just seems like they're more expressed in gonads and brain. But gonads are definitely important because these are the insertions, these are the mutations that get passed down to the next generation. It really doesn't matter what happens in the cells of your of the somatic cells. It's really not that big of a deal, um, but it only affects you. But the changes that are made in the germline affect all descendants. So we kind of want to measure express. I am curious about expression, TE expression um, in germ cells. Anyways, if I'm messing around with this, this data frame is currently easy to manipulate and to be able to draw um, X and Y axis plots. So I can go ahead and draw a plot that compares my mouse and adult juvenile expression. So it's ggplot, that should already be installed. Yep. Dat2, because that's my data frame, I'm assigning my X and Y's. Let's see, I'll put J on the X, J mal expression, and my Y will be A mal expression. And so I'm going to make this a dot plot, geom with geom point. Right, and so these are my data points. These are my X and Y comparison, and generally what it's saying is that more often than not, these expressions, these expression values are correlated. If it's really highly expressed in the juvenile testis, it's also really highly expressed in mouse testis. Although there's definitely some wiggle room around this. Sometimes we'll see TEs more expressed in juvenile testis than we do in adult testis, and that's partially due to the kinds of proteins that are expressed. In juvenile testis, you're more likely to see proteins that do a better job of preventing TE expression. Um, anyways, so a couple of things that we can do with this. So I can recolor this. And so, for example, this doesn't tell me really anything specific. This specific plot doesn't really tell me anything unique. But say I can separate this by color. So I can color code each of my dots based off of the type of the transposable element. So with that, I think will be color is equal to my type column. So my color will be defined by the variables found in my type. That should work. Yes, it does. So all my dots are color coded, all my points are color coded. But if you see the types of TEs in here, the types of elements that are in here, I have a lot of miscellaneous information. So there are some elements that say this DNA question mark. It's a DNA transposable element, although it's not necessarily defined. It looks like one, but we've never seen it before. We're not really sure if it actually is a DNA transposable element. Same thing for like the LTR question mark, other, rolling circle, satellite. These are terms that they're in this data set, but they're not exactly helpful to me because a simple repeat, it is repetitive, but it's not caused by a transposable element. Usually a simple repeat is just caused by weirdness in DNA in the DNA replication process. So I may want to get rid of all those miscellaneous data points. And there are probably a dozen and a half ways to do that. But in my world, how I've kind of figured it out easiest is to use this command subset. Subset. So for example, I may find another variable, I'll call it data sub, data sub. And so I'm going to use my subset. And so how I use subset is to first call subset and then call the data frame that I want to uh, filter that to, and then I'm going to set my criteria. So my criteria, subset is very useful because I can get rid of values that are below a certain number or above a certain number or equal to a certain number. Or a field that's specific to a string character. And so that's what I'm doing now is getting rid of or only um, keeping rows. What I'm going to do as a subset is I'm going to keep a row that has what I want in this type column. So if I don't want to keep simple repeat, I'm going to get rid of that entire row. So I do that by saying my column name type is equal to line. And so here, this probably isn't the first time that I've done this. Is use two equal signs. So in most programming languages, one equal sign is there to assign a variable, but two equal signs is actually checking for equivalency. So you would never say, just for an example, I'm getting just for an example, this isn't part of subset, but I would never say one check if one is equal to one, right? That's more like saying I'm going to create a variable called one that's equal to one. But if I want to say is one equal to one, then I would use two equal signs. It's just kind of a computing syntax. But anyway, so type is equal to line. But I can also add more fields in here, and I can use that by saying or. So I want to keep fields that contain line, or keep fields that contain sign, or keep fields that contain LTR. And so in R, the syntax for or is the pipe, and it's just one pipe. In Bash, it's actually two pipes. In Python, it's the word or and. Um, but in R, at least one of these ways to define or in a conditional statement is to use this pipe. So then I would say, what's my second condition? Or type is equal to sign, or type is equal to LTR, or type is equal to DNA. So these are the four elements that I actually care about that I want to keep in a data set. So I need to spell my stuff right. Subset. Okay. And so whenever I plot redo this plot, but with data sub instead of data two, I should have gotten rid of all those those miscellaneous characters. And so now I'm only comparing expression of my types, of the main types that I actually want to keep, my line signs, LTRs, and DNA. And so this little command, this subset command is really helpful. And again, it makes it's easier. It's in some ways R is better at manipulating data than Python is. So for most FASTA files, anything like that, any kind of wonky looking sequence file, usually Python is the way to go. But for matrices, where we have these columns and row information, usually those R is much better at handling that and filtering stuff out. All right, so I'm not gonna save this as a script for now. I think it's just gonna eat up too much time if I redo some of these things in my script. So I'm just gonna keep working out of the terminal. So if I'm looking at these expression, for sure what we can kind of see already is that signs more often than not are usually the most expressed DNA transpose on type. Uh, that's not that tends to seem to be the pattern, at least in mammals. So if we look at other animals, other lineages of vertebrates, they may not have so many signs abundant. But in mammals, we tend to find that lines and signs are the most common, the most commonly present, most commonly expressed. But I have all these clusters down here. There's a lot of data points in here where expression is less than, say, this metric of 500. So this metric is similar to a TPM. It's like a transcript per million. It's relatively similar to that. So I may actually want to stretch this out so I can look at those data points a little bit better by doing a log transformation. And so I know I have some zeros in here. You cannot transform. You can't do a log transformation of 0. It will give you a non-number. But log of 1 is 0. And there's really not a huge difference between log of 1,000 versus log 1,001. So it's usually not that. It's kind of a common thing to do if you have zeros in a data frame or zeros in a column to just do a log plus 1. So I'm going to do the same thing for my y-axis, log a mouse expression plus 1. And again, this just helps with zeros or handling zeros. All right. So I'm going to, it stretches all this out. Now I can see all of those individual data points that belong to lines, LTRs, and DNA transposons. Okay. But this is muddy. There's a lot of different data points in here. It gets a little hard to look at. There's still some clear patterns in here, like sign is really abundantly expressed. But one field that I have discussed before, but I really haven't explained, is this facet grid. And so this facet grid allows you to separate data based off of a condition. So in this case, I want to separate my data points based off of our x-axis from our y-axis. So if we want to separate these fields based on the y-axis, we would do, say, type is on the y, and everything else is on the x. So if we have to do y squiggly x axis, for whatever reason, you have to define this period at the end of it. So y tilde period. And so that's going to separate these elements based off of the y axis. So now all of these are separated and they're on the y axis. And so it makes looking at these data a little bit easier and a little bit easier to visually compare. But if I want to switch my axes, say I want my be separated on the x-axis. So I want all of my fields to be on the x-axis. I would just do squiggly, my tilde, and then type. So this is kind of why you don't want to use known words to define a variable. So like type is already a function in R. And so since it's already a function, it's popping up with these words that I don't want. But it's just type is defined in my columns. All right. All right, so this makes it a lot easier to compare. So I have them all separated on the x-axis, and so that they all of my variables, all of my plots share the same y-axis. And now I can kind of visually compare expression among all these different elements. At least it's easier to look at this to some degree. So more often than not, remember this is log transformed. So usually my DNA transposons are much less expressed than my LTRs for my lines, and my signs are the most expressed of these types of elements. So again, if I wanted to save this plot, I would just go to export, save as image, make it a PNG, and then save it. Um, and so, whatever, I just saved it. I didn't want to save it. So again, the I guess kind of the last little trick is that these colors can be defined. You can change these colors based off of off of a specific palette. R has a bunch of built-in palettes, but I've gotten a little nerdy enough to make my own palette. Um, so, for example, let's see. Um, this is an album art from a Tool album, Lateralis, and so it has a whole bunch of different colors in it. And so I kind of figured out how to make R separate this image into its entire palette. And so I have all these different colors that I can choose from based off of these scales, these numbers that define a color. Um, so I've created papers where all of my figures are based off of this one palette that's based off of an image, right? So R is pretty fancy. You can get pretty nerdy with it, as I have. But for now, I mean, I don't want to mess around with colors too much because it can get a little bit. You can spend a lot of time just choosing colors. And then especially like choosing colors so that they're compatible with people that are red-green colorblind, right? These believe it or not, these are things that are thought about when making figures. Is how do I want my colors to appear? Will they be friendly to colorblind scientists, etc. But we'll mess around with colors a little bit later. But for now, always just kind of stick to the default and save yourself a little bit of time. But so I think I'm going to call it there now. But we'll manipulate this stuff again on Monday, and next week we might find some. I'll spend the weekend looking around for like public data sets that we can maybe manipulate and play with and graph out. Not necessarily like it doesn't even have to be DNA or genetics based. Just something kind of fun to get better in practice data visualization. But before this hangs up again, I'm going to call it here and kill this stream.